Thanks everyone for tuning in. We're going to start the ABF tools section with our process modeling, TA and LCA work that we have been carrying out in the consortium for the past few years. So this has been a joint effort between NREL and Argonne National Lab. So please feel free to send in any questions, comments to either Tatiana or me if you need them. Next slide, please. So this is just an overview of the workflow that we usually employ in the ABF consortium. We start here in the number 1 with formulating a conceptual process, usually a biorefinery that aims to convert biomass into a specific bioproduct of interest. Then here in 2, you can see we use, whenever available, experimental data to model our bioconversion of mixed sugars to the product or to a specific intermediate, or also to some process separation operations whenever available. And whenever the model is established and ready to go, we run it, and we are able to get all the metrics, all the numbers that are needed to populate the TA and LCA efforts to get many metrics of interest. So for example, the minimum selling price of the product of interest, the GHG emissions, fossil fuel use, water use, and so on. And then we usually feed this information back to researchers, experimental researchers here in 4, so they can understand where they are in terms of actually developing a specific bioconversion route. And then with new data, our model gets updated and we restart the cycle once again. Next slide, please. So as you have seen today from the presentation so far, the ABF consortium is focused mainly on the bioconversion of mixed sugars to specific bioproducts or intermediates of interest as well. So this is just one part. It is the core of our biorefineries, of our simulation work, but it's just one portion of the biorefineries that we need to put together. So to get the full picture, we also need information on feedstock and also on upstream operations that need to deconstruct that biomass into the mixed sugars that we need for the fermentation part of the biorefinery. And these upstream information for these upstream operations come usually from other projects under the BIDO portfolio. And finally, after we do the mixed sugars conversion to a specific product, we have to do downstream processing, so removing that compound from the fermentation broth. In that sense, we connect a lot with other consortia in the BIDO portfolio. So for example, the Bioprocessing Separations Consortium, SEPCON, and whenever needed, we also talk to Chemtech Bio, other projects for catalytic upgrading of the intermediates to the final marketable products. So here on the right-hand side of the slide, you can see just a very simple schematic of how a biorefinery is structured. So going from lignocellulosic biomass, we started with a pretreatment, for example, to get the lignin out. Enzymatic hydrolysis ensues to get the mixed sugars out of the biomass. And then we have microbial conversion to the product, separations, and catalytic hydrogenation or other catalytic conversion processes whenever needed, as we need for meconic acid production and conversion to adipic acid. So next slide, please. So this slide just shows another way of presenting our biorefineries. So pretty much the same operations that I've talked about in the previous slide are here: deacetylation for pretreatment, enzymatic hydrolysis, all the broth conditioning, conversion of sugars and downstream processes to get to the product, but also to get a full picture of an integrated biorefinery, we also need some ancillary operations. So for example, wastewater treatment and combined heat power for production of heat and electricity are also considered. And whenever we have all of this set up, we usually employ one of the benchmark software out there for process simulation, Aspen Plus from AspenTech, to get all the process inputs and outputs that we need, all the list of equipment to populate our TA work, and also all process inputs, outputs, emissions, water, waste streams as well, and to get the full lifecycle inventory and run LCA with the Greek model developed by the Argonne National Lab. So the main purpose of all this is to assess the impact of key fermentation parameters, as ABF is a consortium centered on the conversion of essentially sugars. Obviously, other feedstocks can be considered, but of sugars to some products. And since this is the core of the biorefinery, we just try to understand what is the effect of trimetrics, tidal rate, and yield towards minimum selling price and GHG emissions of several different products. So next slide, please. This is just some examples of some types of assessment that we can carry out in ABF. So one of them initially is covering the metabolic space of interest to ABF, just as a means to provide some information, preliminary information to the researchers whenever needed. So next slide, please, Angie. We have done that for several different molecules. I'm just presenting here two specific molecules, 1,3-butadiene, TA results for 1,3-butadiene, and LCA results for 1,8-cineol. And the idea is just providing this initial guidance to experimental researchers so they can understand where their experimental development of a specific route stands as a function of productivity here on the x-axis and yield, maximum percentage of the maximum theoretical yield on the y-axis. Next, please. This has been shown already at least twice today by Chris and Greg. So we can also, with this type of models, track the progress of these trimetrics over the course of the years. So there have been a lot of progress for the production of muconic and adipic acid. And here at the bottom, you can also see that we can, besides tracking the trimetrics, we can also take a look into the different sugar sources. So for example, different pretreatment methods for the same feedstock, different pretreatment methods, how does that impact the GHG emissions of the adipic acid being produced in this way? So this is has been already published in this paper here. We can send it to Angie. Angie can distribute this later on. And finally, last slide, please. We can also assess different biosynthesis pathways. So this is very important for a few specific molecules. For example, here, malic acid and 3-HP acid. We had several different hosts that could produce these two molecules essentially. And this type of work was very useful in understanding the effect of yield, O2 requirements, nutrient requirements as well in selected fermenting microorganisms. So we have different minimum selling prices on the top, different GHG emissions at the bottom, depending on the host that you use. So this information is also fed back to the researchers so they can understand how to best leverage this type of information for their own process development purposes. So that is essentially it for the TA LCA task.